So I'm going to very quickly go through the sort of few of the work we're doing at CCMI based in Little Cayman. So for those of you who don't know, I'm Jack. I'm a postdoc at CCMI. I actually live on Little Cayman full time. I've been there for 2 years now. And it's an amazing wee rock. If any of you have been there, I'd recommend you get there as quick as you can as well. And one of our many projects that we've got going on is characterizing the biodiversity of offshore seamounts throughout the Cayman Islands. Now, this is truly an international collaboration. We have so many people that didn't make it onto this acknowledgements board to thank, so many volunteers and interns at CCMI who have helped us all the way. But some key partners were the University of Delaware, Dr. Art Trembanis and his lab group, In-Depth Water Sports, if any of you guys know them, Nat, Nico, and Z, and the Smithsonian Tropical Research Institute and the University of Miami were absolutely key for all of our genetic and DNA work, which I will talk to you about. So just a brief overview of coral reefs in general throughout the Cayman Islands. These are what our sort of coral reefs around Grand Cayman and Little Cayman look like in Cayman Brac. They are dominated by these hard corals like Orbicella, these mounding corals that form these structures. And the Cayman Islands in its entirety are completely surrounded by these coral reefs. So this red area here on the map, that is all the coral reef habitats we have. Obviously the sound right here is actually more sand. But in general, you see there's the fringing reef out to the reef wall. And these coral reefs are super important for the Cayman Islands because these islands literally would not exist. I don't know how many of you are residents here or if you were here for the Northwester last February. But this Northwester was absolutely devastating for some parts of Little. This was my back garden when I woke up in the morning. And during the night, the sea actually came all the way up to the house here. And without the fringe reef right out here, breaking those waves, I sort of dread to think how bad the damage would have been. I kind of suspect my whole place would have been washed away because down further west in the island, there were some waves coming 20 feet high and throwing rocks all over the road. Which sort of brings me on to my next point, that these coral reefs, even throughout Cayman, really they're kind of invaluable if you put it in an economic sense. The ecosystem services they provide, such as flood protection, fisheries and tourism, are so important for driving forward our economy, but also for food security. So you think globally nearly 1 billion people live within 100 kilometers of coral reefs. And if you think about some of the lesser economically developed nations like in Southeast Asia, many of these people rely on the fisheries as their actual main source of micronutrients and food. And throughout Cayman, obviously, if any of you guys have been dive instructors, as you know, it's a massive industry here. Most of my friends on Little are obviously dive instructors because it's kind of the only reason you move there. And it's what keeps everyone going. So this is a picture of my friend Bolt on his first ever scuba dive. And I told him he would make it into my presentation. But on top of how valuable they are in like an economic sense, coral reefs are just incredible for their sort of extraordinary biodiversity. So this is a well-quoted statistic that they only cover 0.1% of the ocean floor, so the ocean surface area. Yet they contain 25% of all marine life. So 25% of all species that live in the ocean at some point spend a part of their life stage on coral reefs. And this ranges from your small nudibranchs, which will live there forever, to larger fishes and vertebrates like your sea turtles that will spend juvenile years out on the coral reefs, especially with fish, for example, and then migrate out into the open ocean. But I'm sure you guys are all well aware, if you dive regularly here, the state of coral reefs in the last few years, just throughout Cayman, for example, coral reefs are in a lot of peril. They're facing an absolute barrage of threats from both the local scale and the global scale. And this plot here just shows you an overview of reef functionality throughout the Caribbean. So what I mean by functionality is sort of how useful it is to humans, how much habitat there is to support ecosystem services we need. And you can see if we start at 100% in the 1970s baseline, after we go through these disease epidemics and worldwide bleaching events, we've gone down to about 30% functionality. So overall, we've lost 70% of the functionality that we had on Caribbean coral reefs just in the last 50 years. And some of these local threats are kind of what you'd expect. So you've got your overfishing, pollution, and anchoring. Sea-based sources of pollution were the primary threat in sort of the 60s and 70s. And you can see how somewhere like Discovery Bay in Jamaica or Carrisford Reef in Florida, it used to be fields of these acropora. So that's your staghorn and your elkhorn corals. And then by the time the noughties came round, these entire reefs were just completely gone. So we'd lost all of this habitat, which used to support fish and like little marine critters, juveniles, and it's just become a seascape of algae and rock and sand. But probably the biggest threat to coral reefs, certainly the most predominant threat, in my opinion, is global climate change. You can't look at the news these days without seeing climate change and the impact it's having. Look at the wildfires in California and the hurricanes that were happening last year, a Cat 5, I think it was in June. And the warming climate is really bad for corals because corals live near their sort of maximum thermal threshold. So as it gets warmer, the corals will bleach, they will slowly starve to death, and then they will die. And this is just an overview that up to 2015, we'd had 3 global mass bleaching events on record. And then in 2020, we had a 4th. And in 2023 and 2024, for those of you who are out diving, you'll be well aware, we had a 5th. And the 23-24 one was particularly devastating. So this is an example of some of the diseases we have going on. So this is white band disease, which is exacerbated by the warming climate as well. And this is what wiped out all of our old elkhorn and staghorn sort of coral reefs that we used to have. We've lost about 90% of them just to this disease up to the year 2000. And then here on the left, you have a different disease. This is a novel disease called stony coral tissue loss disease. This disease was first reported in 2014. And unfortunately, it actually kills the corals which seem to survive everything else. So these corals which have survived through all the bleaching and all the other diseases, they're happy as Larry, a new disease has come along. And this has spread like rapid fire throughout the Caribbean. And we certainly have it here in Grand Cayman. If you've seen any brain corals quickly dissolve in a way, it's probably stony coral tissue loss disease. And you can just see how quick the progression is. So in 1 month, you go from a huge brain coral with 5% of its tissue dead to over 60% dead. And then within the next month, that would be completely gone. And this is an example of some of the coral bleaching we had in 2023. So this here is a nice Copperfilia natans. It's a huge, huge brain coral in Little Cayman. And because of how hot it got in 2023, it's completely bleached. It went bright white like this. So this is the coral skeleton you're seeing, and then it slowly starved to death. So this one completely died. And you may recall, if you were diving at that time around here, that the reefs generally look like this. These big Orbiceller bommies, these big mountainous star corals, they were just bright white. You could see it from the boat. You would be going over a reef, and it would just be a white glow coming from it. So 2023 was a horrendously warm year. And this is sort of the damage it did to Little Cayman. So at the start of 2023, we were sitting very comfortably at around 25% coral cover on our reef. So that's quite high compared to the rest of the Caribbean, a lot higher than Grand Cayman as well, because obviously we're an isolated island, small and remote. We don't have any land-based sources of pollution. But because of the bleaching, by the end of 2023, January 2024 right here, over 55% of all of our corals were killed by the marine heat wave. And this reduced the coral cover from our very stable 25% down to a new record low in Little Cayman of 9%. However, this sort of brings us onto this project where depth can be refuge for corals. So this bleaching is caused by the interaction between light and temperature, so sunlight and warmer seas. But as you sort of move down beyond 30 meters into the mesophotic zone, so 30 meters is what, 100 feet? you find these reductions in temperature and light attenuation. So that means some corals are able to survive down there, no problem, while there is a marine heat wave and a bleaching event going on. But the only caveat with this is obviously you'll have different species, and occasionally, quite often we find these species are actually more sensitive. So it's sort of a trade-off between will they be refuge rather than can they be refuge. So sometimes, the right circumstances, yes. So we undertook this project. The only sort of big problem with studying mesophotic reefs is it can be a bit of a challenge sometimes, because when you're beyond 30 meters, 100 feet, that's often beyond the recreational dive limits. I think the PADI limit is 100 feet. So we have to have specialist training for our tech diving team, specialist training and specialist equipment. So this involves using either closed circuit rebreathers or open circuit tech methods where you carry your stages down so you can spend a long time on the bottom, you know, at 150 feet or so, and then you slowly work your way up with decompression to make sure you don't explode from nitrogen. So what did we find on the mesophotic reefs in Grand Cayman? Well, really our fish families that we modeled biomass for were actually the same or very similar to the shallow reefs, the shallow water reefs throughout the island. And the benefit cover was the same too. So we had a complete dominance of macroalgae beyond 30 meters, just like you do in the shallow waters in Grand, and really very low hard coral cover, only about 5 or 6%. And this is to be expected if you consider sort of how close we are to the island. So these were the 4 sites where we undertook our mesophotic surveys. I'm sure many of you guys have dived them. I don't dive much in Grand. I do all my diving in Little. But you can see just how close they are to this sort of hugely Georgetown and West Bay, such a built-up area. So the stresses are probably still impacting these mesophotic reefs in Grand. But then what about seamounts? So seamounts, they're underwater mountains. They come up at least 1,000 meters above the sea floor, which is the same definition as for a mountain on land. It has to be 1,000 feet above the base. I don't know why, or 1,000 meters, sorry. I don't know why we choose 1,000 meters, but a lot of stuff in science is arbitrary. And these habitats are super important because you think about it, you're in a desert in the wide ocean, and then suddenly you've got this huge pinnacle rising up where sessile organisms can sort of settle on, and then your motile organisms, your fish, can come in and find habitat and find food. They're likely very important for genetic connectivity. So you have these seamounts in an ocean, they're stepping stones for organisms. And they massively influence nutrient cycling. So you think you have a mountain coming out of nowhere and some currents coming through, you get some upwelling, which moves things around and often makes seamounts quite nice fishing grounds. So where are these seamounts in the Cayman Islands? You guys are probably all well aware of this, but there's 12 Mile Bank, which is just 12 miles west of Grand Cayman. And there's Pickle Bank up here, which is about 50 miles north of Little Cayman. And like I said, they're really, really great fishing grounds. They're great fun. I'd recommend everyone tries to make it out to either of these seamounts whenever you can. Even if you don't want to dive, if you just want to go fishing, you should absolutely do it because it's a great place. It's a great place to hang out. But what's kind of really surprising to me is these ecological communities on these seamounts, before we started this project, were never characterized. No one ever went down in scuba gear and did in situ surveys to actually document what was there. So any stuff we knew was there was from recreational diving or from fisheries data, so taking a boat out and just seeing what you caught in your nets. So what we aim to do with this project was to characterize these communities with in situ surveys. So send our scuba divers down, they'll take down their transect tapes and their quadrats, and they will collect the data that we need to characterize these communities, and also map each seamount. So these are with our boys from the University of Delaware. They had some sonar technology that we took out to the reefs each time. And then scale up diversity. So what I mean by scale up diversity is we can collect environmental DNA from the sediment and the water to capture anything that we don't see on our traditional surveys. And also, we collaborated with some colleagues from the University of Miami, Harbor Branch, to inspect genetic connectivity of seamounts throughout the Cayman Islands in general. So the things we needed, firstly, we needed a vessel. We chartered El Puma, so this is in-depth water sports. And we took their crew for a few weeks each time. I think we had them for a total of 4 weeks over the last 2.5 years. So this is Z in the circle, Nat who owns it, and Nico, good bunch of lads. And we had some emotional support animals as well, because when you spend a week with colleagues on a boat, you don't like them in the end. You need a cuddle with someone. So for site selection, we first would send out our mapping team. So these are the guys who do physics or geography or whatever, and they make one of these really hugely impressive side scan maps. So this is from sonar imaging, and the resolution is less than a meter. So you can see this bright red or orange ridge here. That's the shallowest point of the seamount. That's at about 30 meters depth. And then you can see the wall dropping down here beyond 70 meters. So they got an amazing, amazing detail for us to choose where we could go dive in and sort of characterize the communities. And then we would send in our tech dive team. So these are many of our tech divers. I think I've included all of them. You've got Ashley, Matt, Alex, Gretchen, and Ryan. And then this is the general sampling team. So these are the guys who were on the boat and would process samples, collect samples from the sea, and then take them back to the lab with some very late nights. This is Dr. Mathieu Leray here. He's from the Smithsonian Tropical Research Institute, and he's fronting all of our environmental DNA work. So that will be amazing when we finally get that back. So 12-mile bank. Just a reminder of where it is, just 12 miles west. So we went from West Bay up there where Nat was based. And after producing the side scan map and choosing our sites, we could finally hop into the water, and the rest of us would just wait on the boat like this while they were down there for an hour or so collecting their data. And this is how we collected the benthic data. So I just want to talk you through this quickly. This is just some PVC pipe with some GoPros and lights on. And what you can do to collect benthic data, rather than spending the hours down there counting everything you see, you get a couple cameras, you go 1 meter over the reef, and you can cover over 100 square meters in one dive. And then you take it back up and you can stitch it together to make some software that you can then analyze ex situ, so on land afterwards, after you've collected the data, which allows us to collect so much more data. And then these are your traditional fish surveys. So we would send someone down with a rebreather, they would take a transect tape out and just record all the fish you see and the size of them. And this is sort of an example of how you collect eDNA from the water. So a whirl pack is a fancy Ziploc bag that they charge you 10 times more for, and you just collect seawater in, get it on ice, then you can process the DNA from it. But we did have some rough conditions, and I am really sad that I don't have a video of this. But this is one of our open circuit tech divers, and he's on top of the seamount right here on a sandy patch and rocks. If you can see his bubbles up here, they are absolutely ripping from the currents. So this gives you an idea of how sort of rough it was on some of the days out there. It would look fine on the surface, and you would get down, and these currents would just blow you right off. So that was a learning experience. And the next time we went, we took these DPVs, so our scooters, to make life a lot easier and make sure no one gets bendy. But what did we find on 12-mile bank? Well, it was sort of really the same fish families as we found in Grand Cayman and what you expect on shallow water reefs. It was your damselfish, your chromis, your wrasse. These are the little things you see hiding in the nooks and crannies and what make the coral reefs look so beautiful. But we did find a nice clear north versus south difference. So you can see that the north side, we found a lot higher density of fish, a lot more fish, more diversity and more biomass. And this is probably driven by sort of differences we have in the topography and the benthic community on 12-mile bank. So you have more fish on the north side and the south side because of the benthic cover we have and what lives there, so more corals and things like that. And also the sort of hydrodynamics, so where the upwelling's coming from and where fish would like to hang out. So this is an example of the photomosaics that Alex there, one of our rebreather divers, collected. So this isn't a raw photo. These are 3D models of the benthic community. So after you've stitched them all together in Agisoft software, it spits out the highest detail. Obviously, like there's a gap here because there's not been enough overlap of the photos, but you can see really well just with 2 GoPros, so quite cheap. You can see very clearly that, that's a Paraides asterides. We know what coral that is. You can ID that coral, and then you can see the benthic community. So after we commandeered loads of volunteers, which is why the acknowledgements list is so long, you can have people identify what we found down there. And then with the environmental DNA from 12-mile bank, unfortunately, we don't have those data to hand. They're still being extracted. But this will allow us to detect any cryptic species that you wouldn't traditionally find on a survey. But also, with our survey method, it'll allow us to sort of find any of the large pelagics that we don't see or that see us first and then swim away before we get down there. So next, I'll take you through to Picklebank. So just a reminder, Picklebank is about 50 miles north of Little Cayman. And our original plan was to live out there for a week on a working vessel. But I can't remember exactly what happened. But someone ran aground in the Brack, I think, or hit the reef. And our vessel was then stuck in the harbor in the Brack. So it couldn't make it out. But to the rescue, we had these boys again. They really are heroes, and I would dread to think how this project went without them. So in general, going to Pickle, it was an amazing experience. I'm going to emphasize, guys, get out there if you can, like for a dive or fishing. What great fun it is. It was generally very chill. Sometimes it was a bit choppy, but more often than not, it was like that. Beautiful views. And then I don't know if this video works. We even have a video.
What month were you there? What month?
July 24. Yeah. And if anyone knows their dolphins in the Cayman Islands, and you can tell me what that is, I'll buy you a pint. Because so that was our general commute in the morning out to work, which was really nice. But first we would send our mapping boys out. This was from day one. They produced a very nice preliminary map for us. It was really cool to see this in action. And eventually, this map isn't final. We don't have a final product yet. But the resolution, because we managed to have Dr. Art Trembanis out with us this time, the resolution and the detail they managed to go into for producing this map of Picklebank it was just incredible for helping us choose our sites. You can see ridges and troughs running through, so spur and groove on a bank itself, which is absolutely incredible. And then it was time to dive. So we would go down, collect our fish data as before, and our eDNA. But we didn't actually collect any benthic data from Picklebank because of the conditions with the currents out there and sort of the logistics. We had a few divers having some ear problems and things like that. We decided not to collect benthic data. But I wanted to show off this pillar coral we did find on Picklebank, which is really quite incredible because this is a critically endangered and evolutionary distinct species. So pillar corals have been wiped out throughout a lot of places in the Caribbean. They get absolutely hammered by disease. We only have 10 left in Little Cayman itself, which is, a very well protected and isolated island. So I don't know if there are any in Grand still. You guys will know Grand better than me. So to find one out on the bank with no disease, most importantly, that was a really good sign of the benthic condition there. So what do we find here? Well, reef fish sort of dominated again, so your parrotfish and your wrasse. But we did also find quite a high biomass of your almost fishery target species. So I personally love snapper. We found loads of snapper and loads of jacks, horse eyes, yellows. whatever else. But in general, there were a lot more carnivores we found at Pickle Bank, a higher carnivore biomass compared to 12 Mile Bank, and then also compared to Grand Cayman. We also found a clear depth partitioning in the community. So on Pickle Bank, we had the opportunity to survey the reef at the shallowest depth of 25 meters. So it does come up really shallow given its location in the Caribbean Sea. And we found really distinct communities between the shallow reef and the deep reef at 45 meters. And this was primarily driven by the presence of these fairy basslets and these small chromis which exist in the shallow waters but don't like the deeper waters. So we spent some long days out on the boat sampling. We would come back late at night, sunset, that's late at night for me, around 6 p.m. on the dock after a hard day's work. And then we would be up in the lab late at night processing some samples. So we have a rule as well with fieldwork, that fieldwork is tiring. And there were some victims who weren't used to this type of work, falling asleep around here. This beanbag actually took many victims, an amazing beanbag. I'm glad they brought it out. And my poor intern, unfortunately, didn't get the memo that you don't fall asleep on the job, because you will be bullied for it. So what are our next steps from this project? So just to quickly recap, all these locations are generally dominated by reef fish, which is a good finding. It's what we expected. But we found more macrocarnivores. So your fishery target species, the further we moved away from the population hub. So the further we moved away from Grand, we got more carnivores. And then, so 12 Mile Bank had more than Grand, and then Pickle Bank had more than 12 Mile. And this makes sense, right? With the lack of fishing pressure at Pickle Bank compared to 12 Mile. While we were out at Pickle for the week, I don't think we saw a single vessel while we were there. The only fishing presence was probably us. And still to come will be the insights we get from our eDNA analyses. So Matthew Leray will lead that. And the same with the genetic connectivity. So this type of analysis takes lots of time and unfortunately requires a lot of people to coordinate their time to work together and extract the things we need. So if we think about the big picture of why do we do this work and what's to come of our work, firstly, why do we do this? Well, there are some massive threats to offshore seamounts, and these threats are going to only get worse in the future. One of these huge big threats is obviously fishing and destructive fishing practices. So things like your trawling, they can completely alter the motile community, and if you use destructive fishing practices, you can actually destroy the benthos itself. But as with seamounts and mesophotic reefs, the sort of impending stressor, the impending doom of climate change So we did this work because if we want to manage these future stressors, the fishing and climate change that's coming up, we need to have the basis of the ecological data to inform any management strategies. So what's to come of our work? Firstly, we're going to contribute to the Biodiversity Action Plan. We have drafted a chapter for seamounts for the Cayman Islands National Biodiversity Action Plan. Before we drafted this chapter, it did not exist. The Cayman Islands did not have one for their seamounts. So that will be really nice when we submit that to the Department of Environment. And this opens the door for any sort of red listing activities to initiate any conservation action. And also, these data can form the basis for any implementation of your marine parks. So how we can manage these environments to mitigate any damage from fishing if fishing is to go on there. But just a quick note on marine parks. Fishing is incredibly important. I'm a fisherman myself. I love fishing. Going out catching snapper is my favorite. This is from Salt Rock Dock in Little Cayman. This was actually an awful night. I didn't catch a single one of these. My mate caught all of them. And this is out from the wall. You can catch his tuna and things like that. So it's super important that we accept fishing is going to happen and that we encourage it to happen sustainably. So management for fisheries needs to be flexible and also match the needs of society. We can't just ban fishing completely because you take away people's livelihoods and you also take away a vital source of nutrients for humans. And something like a marine park, while it can be a tool under climate change, it's not going to stop climate change. You can't just put a marine park down and say, this reef is safe now. So it has to be made sensibly with clear goals in mind. And our future work really will just be to hopefully get back out to some of these seamounts with some future grants and have a more holistic characterization of the entire community of both the benthic and the fish. So for example, we would love to go back to Picklebank and actually quantify the benthic community there. And we're going to continue to investigate the role of these seamounts, so both the mesophotic aspect and the isolation aspect for refuge for corals and fish while the climate continues to get warmer. And this also allows us to answer some academic questions such as about assembly rules of fish, which are exciting to some people but not to others. So my sort of key messages for you are these mesophotic offshore seamounts are really some incredible ecosystems. They're very unique. And if you can go out diving to 12 Mile, absolutely go for it. And if you can go out diving or fishing at Pickle, you really, really should, while it looks as good as it does. And our work will hopefully contribute to their future management to be able to sustain them for a bit longer. But this really is just a first step. There is so much more we can do to investigate these seamounts. It just requires a huge effort and a lot of time. So this is the core crew from the final day at Picklebank. These are the survivors, the ones who made it. So huge thanks to them for all their work on this project. And thank you guys all for listening.